Okay, so we have a great speaker today, Christiana Pettibone. She has a PhD and almost 20 years experience managing, directing, and guiding public health policy research and evaluation projects. She holds a PhD in policy sciences from the University of Maryland, Baltimore County. Prior to joining NIEHS, she served as director of the Mayo Tech Corporation Center for Community Prevention and Treatment Research. Kristi has worked on evaluations of several federal grant portfolios, including CDC's Injury Control Research Centers and NIEHS's Environmental Health Core Centers program. For the last 3 years, she has led an NIEHS work group in the development of a new translational research framework. Kristi, we're so excited to have you here. Please go ahead and start your presentation. Thank you.
Great, thank you. And I just realized that I didn't ask for information about how to move the slides.
You just ask and I will move.
Okay, great, sounds good. All right, so you can go ahead and go to the next slide. I am with the National Institute of Environmental Health Science. We're the only institute that is not up in Bethesda. And I'm in what we call the Program Analysis Branch within the Division of Extramural Research and Training. My formal title is Health Scientist Administrator, but I call myself an evaluator, and you'll see why that's important as we move on. But my primary responsibilities are to conduct program evaluations with the twin goals of identifying areas for program improvement and communicating accomplishments and discoveries of our grantees. So I don't know how much you guys know about the NIEHS mission, but it's to discover how the environment affects people in order to promote healthier lives. So because most of our work focuses on reducing harmful environmental exposures, many of our grantees' accomplishments and discoveries actually result in prevention work rather than drug development. The translational research framework that I'm going to show you is a new model that we are rolling out that really helps make a place for environmental health science in this concept, within this concept of translational research. Our hope is really that this model helps us drive change and gets our grantees to those public health impacts quicker. So you can go to the next slide. So we like to think of the translational research framework as a map. And so you'll see this style slide kind of throughout the presentation. And I'm really just sort of using this as the overview of the presentation. So I'm going to talk a little bit about the evolution of translational research at NIEHS. And then I'm going to go over what we think of as the 4 innovations that we have made with this framework over previous translational research models. Then we'll dig into a little bit of the details of the framework so you can understand how it works. And then I'll talk about how we actually use it to evaluate translational research. And then we'll talk about some of the challenges that we have with it and some of our next steps. And hopefully there'll be time for you guys to give us some input about what you think about it and if you're having some of the same challenges. So you can go to the next slide. So first, the first section here, we'll talk about the evolution of the translational research at NIEHS. And you can do the next slide. We've really been working on developing this framework for the last 3 years. And we finally published an article on the framework this summer. And you can find it if you look at the July issue of the Environmental Health Perspectives. It's kind of hard to give a link, a hyperlink on a webinar like this, but you can get to it that way. After being focused on it and sort of being in the weeds with it for 3 years, though, as we started rolling it out, I really had to stop and kind of come back up to that 30,000 foot level and ask myself, why are we doing this? What is it that we're really trying to accomplish? And when I stopped to sort of think about that again and just revisit it, it really comes back to that we're really trying to understand the big picture impacts of investments in fundamental research with the purpose of really being able to communicate the value of biomedical research and really be champions of the science. And then finally, working on this framework is it really allowed us to define translational research and provide a structure and a common language for both our researchers, but also our staff, so both our grantees and our staff. And with this common language and this common understanding of the translational research categories and the idea of translational research, and having that as a really concrete framework, it allows us to do a better job of evaluating the impact of these translational research ideas. So you can go to the next slide. So before we get too far into it, I do want to take a look at the definition that we use for translational research. And we really just consider it the evolution of a research idea into concrete strategies that protect and improve human health. And this definition is worded in such a way as to avoid any kind of directionality or linearity. And then we also tried to frame it in a way that was general enough that it could be applied to areas beyond the environmental health and biomedical fields. Go to the next slide. So this is just a quick image, just to kind of establish that there is a long history of translational research and the concept of translational research. And this is really to just kind of show how it's evolved over the last 20 years or so, from kind of being a concept in that first row of, T1, T2, sort of basic to clinical, through to more recent evolutions of translational research that factor in more public health work. You can go to the next slide. As the concept of translational research became more concrete, it really gained traction within the NIH community as well. And in 2012, NIH launched an entire institute devoted to translational research, the National Center for Advancing Translational Sciences. The diagram shown here illustrates their model of translational research, and it includes the 5 different nodes that symbolize the different kinds of translational research activities. Within the NCATS model, translational research is similar to ours. It's really looked at as being sort of the transition of an idea from sort of one of these circles or nodes to another. And the lines really indicate that there is no linearity, that things can really sort of go, it's multi-directional. As you can see, this model really puts patients at the center, indicating that their involvement is everywhere. And then you can really also see that 3 of the 5 nodes are specifically focused on clinical activities. So on the next slide, there we go. The evolution of translational research at NIEHS has really sort of been going on in parallel, in that we have lots of programs that support and facilitate translational research and that are sort of tasked with doing translational research. The NIEHS Core Centers program is one of our flagship programs, and as such, we conduct evaluations of it about every 5 years or so. And during the last evaluation that we did, we focused on assessing the center's support of translational research. To do this, we used the NCATS model, and it's a really solid framework that's especially relevant for clinical treatments. But if you remember, when I mentioned a lot of the work that our grantees do actually results in things that are not clinical. They're really working more in sort of prevention fields and public health fields, so they don't tend to have so many clinical impacts. So when we tried to apply the NCATS framework to our evaluation of these core centers programs, it really fell short. So I want to tell you a little bit about the evaluation just so you can understand how we started down this path. We started off asking our grantees in the Core Centers program to send us narratives, to send us examples of research that they thought was translational. We also read through the progress reports that they send us every year, and we identified examples that they put in their progress reports that we thought were translational. That we read through them and said, "Yep, this qualifies. This really shows an evolution of an idea over time." So we ended up with about 200 research narratives from the NIEHS Core Centers. Excuse me. Within so there's sort of 2 different components. So there's the research narrative, and that's the full story. And then there is the research milestones. So one narrative might have 2 or 3 different milestones in it. So we had I think we had 208 narratives and about 241 specific milestones within those narratives that we reviewed and coded. And we use the CTSA Translational Research Framework. They have developed a checklist that allows you to kind of go through and figure out exactly which area or category of translational research a particular idea falls into. And they are actually using that to sort of look at and code publications. And so we sort of translated that and used that for the narratives that we have. And excuse me just a second, I have a cough. Oh, sorry about that. So you can go to the next slide. When we did the evaluation, the first thing we did was sort of bin everything, code all of those translational milestones into one of the 5 categories. And when we did that, we found that about 85% of all of the coded milestones fell into the basic science category. So that was sort of our effort to categorize each of the milestones. But if you think back to our definition, what we really are interested in when we think about translational research is the movement of an idea or the bridging of an idea from one area to another. So if you go to the next slide, the next thing we did was to look at how many of those larger narratives included one or more milestones or one or more research areas. But when we did this, we still found only about 86% of those narratives included more than one translational area. So there wasn't even a whole lot of bridging going on. There wasn't a whole lot of movement out of these that basic science area. All right, so you can go to the next slide. So when we discussed these results with our program staff and with the evaluation advisory committee that was giving us some input on the evaluation itself, this is the reaction that we got. Everybody felt really strongly that these findings didn't adequately represent the true translational nature of the work that the core centers do. So you can go to the next slide. And in reflecting and discussing these findings, we identified 4 challenges that we thought created a mismatch between what we all thought the centers did to support translational research and what the evaluation findings actually showed us. So you can go to the next slide. So over the course of 3 different funding opportunity announcements, or RFAs, NIEHS told the core centers that they should support translational research, but we never really provided a clear definition or concept of what we meant by translational research. So that kind of caused a bit of an issue. Second, there's so many types of research that are lumped into the NCATS basic research category. It was really difficult to capture the nuance of the basic science research that our grantees do, and to be able to recognize the handoffs that occur within that basic science area as translational. Third, the NCATS model, as I pointed out before, is really focused on clinical translation, and it's not designed to capture the type of prevention and policy impacts that NIEHS grantees tend to work towards. And then finally, this was, I think, a little more on us and sort of the evaluation, that the data that we used were these narratives. And as I said, probably half of them, maybe a little more, came from their progress reports, which are designed to sort of tell about work that they have done over the course of a year. We were able to pull some of them from their 5-year annual reports, but even those, they tended to just copy and paste things from their progress reports. So that wasn't a really useful way to capture sort of a long-term narrative. So the way we thought about this is, what we did was to capture snapshots in time of their research. So we kind of caught one little picture, when what we really wanted was a movie. We wanted to be able to sort of capture the dynamic nature of this research and its evolution over time. So you can go to the next slide. So when you work at NIH and you have a problem like this, usually you're asked to form a work group. So we formed a work group. We had representatives from every branch in our division, as well as representatives from intramural research and our national toxicology program. And we kind of worked through a whole bunch of translational examples and tried to identify those components or those concepts that that we thought provided evidence of their translation. And from there, we sort of developed and crafted a new idea of translational research. And when we finished that, we took it around and we got feedback from folks at other ICs, from some of the NCATS, their Clinical Translational Science Award evaluators, from CDC, and also from our grantees. So you can go to the next slide. So as we were going through this process, I happened to be reading this book. So I don't know how many of you have heard of it or read it, but it's over 500 pages long, and it tracks the history of the gene from Mendel's work in 1865 to our more recent efforts mapping the human genome and editing specific genes. And I know I looked at it and I was like, oh my God, there's no way, that's got to be super boring. It was an amazing book. I read it on vacation and it was fantastic. So I highly recommend it. But I was reading this book as we were working on this translational research framework. And you can go to the next slide. In the back of the book is a timeline of key points or milestones along that research process. So from Mendel's findings in 1865 to our current work with the human genome, it's all mapped here. But if we used the NCATS model to categorize all of this work, everything here would get binned as basic research. And if we consider translational research to be the movement or the bridging of an idea from one translational area to another, then even this 150 years of groundbreaking work, if we use the NCATS model, it still wouldn't be considered translational. And that didn't seem right. So you can go to the next slide. This is just my reminder to myself that we're switching gears. And so the next piece is that I want to give you a little bit of an overview of sort of how NIEHS began to focus on this concept and describe the actual innovations that we've made. So you can go to the next slide. So the 4 innovations of the framework, on the left, we've got the NCATS framework that we've talked about before with the 5 areas. You can go to the next slide. The first innovation that we made was to change the orientation of the graphic to represent more of a 3D model. We've got lots of chemists on our staff, and so lots of folks like to say we were sort of influenced by atomic structure, possibly solar system models. I like to joke, you can go to the next slide, that I was more influenced by a certain retailer. The next slide pops up. So you can go to the next slide. But basically, our orientation, we created 5 rings that represent the major translational research functions. And so we used what I sort of consider a target model, where we've got these 5 rings. And you can think of each of the rings as being equivalent to the 5 nodes on the NCATS model. But more importantly, we reframed the framework to use language that recognizes the contribution of non-clinical work to the translational process. So the 5 terms that we used for the rings really tried to avoid anything that focused on the clinical work. But as you can see, you could look at this and it doesn't even really focus on environmental health or public health either. But when you sort of step back and next slide, sort of look at a view of the full model, it can be overwhelming. But the main point is that the second innovation we made was that along each ring, we've added nodes that describe the activities that take place as part of that type of work. So this is where we built in more of a focus on environmental health science research. But we also think that this model could be adapted for other fields. We don't think that these nodes are exhaustive, but we have used this framework to map out about 20 case studies so far. And so far, we've been able to classify all of the translational research activities using these nodes. So it's working for us. But like I said, right, another field could take this and fill in nodes that work for them. So the next innovation, you go to the next slide. The third innovation is what we consider sort of a more dynamic way to describe translational work in the basic science area. So, when we used the NCATS model, we found that everything got lumped into that basic science category. And so we really wanted to come up with a way to address that challenge and be able to capture more of the granularity in that area. So we really focused a lot of thought and effort in this, what we call fundamental questions ring. And what we have found and what we have proposed in the model is that there's really three components of this fundamental questions ring. So there's the research drivers, the experimental settings, and the organisms. So I'm going to walk through sort of each of those three components and just briefly describe what we mean. So in looking at the examples of fundamental research, we discovered that all of the examples tended to focus on three main research drivers: identification, observation, and understanding. So the identification is really asking, what is this? What is it that we're seeing? The observation is, what is it doing? What are we observing? What's going on? And the understanding is, how does it do that? So that's where we can really get at, sort of some of the mechanistic understanding that's going on, but also, some of the behavioral understanding of why are things happening. So if your research addresses one of those questions, it belongs in that fundamental questions ring. The second component of the fundamental questions area is the experimental settings. And the 6 nodes just represent sort of the 6 primary experimental settings into which fundamental research tends to fall. And we just like to emphasize that similar to the NCATS model, we recognize that there is epidemiological research as well as the sort of basic lab research that are both considered part of the fundamental questions ring. So the third component, you can go to the next slide, is organisms. And so we also think that the organism being studied matters. We just kind of put up a couple different examples of what some organisms might be. It's sort of an endless list, though. So you can go to the next slide. This is how we represented it in the 3 components all together as one unit in the framework. And you can go to the next slide. So to talk about the 4th innovation, I just want to revisit the full framework again. So in previous models and previous ideas of translational research, a research idea had to move from one area or one ring to count as translational. And we posit that moving around the ring from one node to another also represents a translational bridge. Each node represents areas of work that require different tools, backgrounds, training, different experience, different people, different resources. And as such, moving from one node to another really does represent the bridging of an idea from one area to another. So the next slide, we look at an example of this so that you can kind of maybe explain, so it hopefully explains the idea a little bit more. In this example, which is looking at the effect of pesticides on human health, the first milestone is comprised of animal studies that were conducted to understand the health effects of pesticides in animal models. And then in milestone 2, other researchers were sort of looking at the epidemiological data and were observing connections between parents who were working in the agricultural fields and childhood cancer for their children. Using the NCATS framework, both of these milestones would have been lumped into basic science, and we agree that they are both fundamental biomedical research studies. But the resources, tools, training, and people that work on these 2 types of questions are different. And as such, we recognize, and our translational research framework recognizes that the connection between these 2 areas represents a translational bridge. But you can go to the next slide. I just kind of throw this up here to just reinforce why we think it's important to recognize movement around a ring as translational. And as NIH and NIEHS are increasingly emphasizing team science and interdisciplinary work, it becomes more and more important for folks to be able to work together and for there to be multiple team players. And I just like to use the example of soccer. Both my kids play, and the goalie doesn't do all the work herself. If you know soccer, right? She's got a defensive line that is helping her defend the goal, and then there's other people who are moving the ball forward. So it does take a team, and each person has a different role that they can play. And the same thing is true in science. This idea might require our grantees to make partnerships, to work with communities, to form these partnerships. what we say is that, right, they don't have to be able to do all of this work themselves, but they should be aware of the different areas, so that they can be aware of what they don't know, and they can be aware of sort of identifying people that they know that can help them with this work. So you can go to the next slide, and we'll move on to the third area of the presentation. And we'll just do sort of a little bit of a dive into the other rings of the translational research framework. So this second ring is what we call the application and synthesis ring. And this is where researchers conduct experiments in a structured and predictable setting to gain a deeper understanding of a process or a particular effect. The types of activities that are included in this field might include pilot tests of interventions, testing of new methods or approaches, new tools, or testing of other types of ideas in very highly controlled settings. So the next ring is about implementing activities in real-world settings and adjusting those activities to account for differences in different settings and populations. So we call this the implementation and adjustment ring. And examples here might include things like validation of biomarkers and screening tools, clinical testing that you're doing in the real world, validating tools, looking at sort of effectiveness and value analysis. Did we? I think we may have actually skipped a ring. If you go, yeah, there we go. So that's the implementation and adjustment ring. The fourth ring is the practice ring, and it focuses on moving established ideas into common practice. This can include using evidence to inform new clinical guidelines or biomedical approaches. So we don't do away with that clinical component altogether. It is still a critical piece of translational research, but it also provides a place to recognize that the evidence may be used to develop public health practice interventions, to develop policies, those kind of things. And the idea here is that, once you've gone through the first three rings, you've really developed, you've defined the evidence, you have established that your idea is solid, and it has gained enough credibility to move into the sort of common practice. So this is where we see things moving into common practice, and being in a place to affect sort of a broader population health. And finally, we have the fifth outer ring, which addresses impacts. This is where we assess the broader environmental impact the broader impact of a particular practice, guideline, or policy that was sort of put in place in that fourth ring. So this is where, for example, a state might implement a policy to reduce air pollution. Researchers might want to then continue their research to see what the impact of that policy is on air quality or on related human health outcomes such as lung function or asthma rates. So this ring is really looking at, okay, you did you had your research idea, you tested it, you demonstrated that it was an effective, improvement over, previous ideas, and it sort of got to be accepted into the common practice. And now you're saying, okay, do we actually see changes in what we wanted to see once it is in common practice? And the thought being that based on what you find in that assessment, you can go back and revisit. So that covers the whole framework, and you can find more details and resources about the translational research framework and about other translational research activities at NIEHS at the niehs.nih.gov/translation. But I want to make a few more points about translational research, and you can go to the next slide. So the first caveat about translational research is that we recognize that not all research will be translational. The NIEHS mission is to discover how the environment affects people, though. So while there might be work going on in the lab, we really want people to think about, even if they're not doing their work themselves, about how their work can be used to have an impact on human health. Our hope is that this framework starts to help our grantees think about that path a little bit more concretely. The next slide. We know that not all translational research is linear. But to continue to be a champion of fundamental research, we need to be able to tell coherent stories of how these investments in fundamental research actually lead to human health impacts. So we recognize that translational stories can start at any node or ring on the framework, and really go to any other node or ring. And it is a messy process. As you'll see, we've got an example that we map out, and you can see that it bounces around. So we'll see that in just a minute. The third caveat is that we know that serendipity plays a part in translational research and that you can't predict it. But we also think that we can do a better job as science managers at teasing out what exactly serendipity is and about nudging advances forward by mapping out work using this framework. I will say that we just had a group of our children's environmental health grantees present their work using this framework. And several of them sort of talked about the role of serendipity and sort of just being in the right place at the right time. And, one of the arguments, though, is that you can have that luck, but you also have to have the preparation and the knowledge and the openness to be receptive to that luck and to, maybe an unintended finding. But that sort of knowing the path that a research idea might take through this translational framework can put you in the mind to think about where that idea might go in the future. So next slide, to stay with the storybook theme, we also recognize that translational research takes a long time. It's not something that we're going to see over the course of a year or two. It can be something that may take 20 years. So next slide, you see my theme here. So I just kind of want to give your brain just a break for a second from processing all of that information. I don't know if I've still got a little bit more to go through. I don't know if people have one or two questions. We could take a question now, or we can keep going. So,
Any questions for now?
All right, well, let me go ahead and go on to the next section then. And this is looking at how we are using the framework to evaluate translational research at NIEHS. So in an ideal world, and you can go to the next slide, a translational bridge would look like this. It would be a nice, clean, straight path from one starting point to the other, to the end. But if you go to the next slide, we all know that translational research looks much more like this. It's messy and it's chaotic. And that's why being able to map and track and evaluate translational research is really fairly challenging. So the next slide, just to revisit the title of the talk, because it's sort of driving change and to add to the purpose of why we're doing this. One of the main reasons that we are focused on evaluating translational research, one of the several reasons that we're focused on it, is one is so that we can identify opportunities for these translational bridges or handoffs. And whether that's us as science managers identifying those opportunities or facilitating our grantees being able to identify those opportunities. It's sort of being able to identify those and help them happen even more effectively, whether that's faster or more successfully. The second piece is that by looking to see where researchers are successful in doing translational research, we can try to identify the strategies that they're using and the things that are helping them do translational research well and try to replicate those things. And then on the flip side of that, we can look to see where researchers are having a hard time moving their research out to impacts, and we can try to find ways to help them navigate the translational research process. So with this new framework in place, we followed the lead of the evaluators at the CTSA program, the NCATS program. They had developed this checklist that we had used in our evaluation of the Core Centers program. We took the checklist and we adapted it to fit our new framework. We're still fine-tuning this. It is available on our webpage, or actually I think it will be available in the next few days. But it is, so it's still a work in progress, but a couple of the things that we did differently than the original checklist that NCATS had put out is that we tried to put some quantification in here. So we added the little sections that sort of let us say, all right, how many of these sort of fundamental questions categories are there, and tried to account for that in this checklist. But it's still not a really elegant way to track or count bridging around the ring. So you can go to the next slide. And so this is what it looks like if we map out sort of a select set of translational milestones using the translational research imagery that we've come up with. This imagery really helps us see the movement of an idea from one ring or one node to another. And I can walk through this real quickly, but again, this is going back to the pesticide research and what we call the take-home pathway. So farm workers who are working with pesticides out in the field go home, change their clothes at home, and pesticides that have kind of clung on to their clothing then get transferred to the home, into the carpet, into the wash, and then the children come in contact with that. And so, when we map out the evolution of this research over time, it does start off with an observation in, sort of an epidemiological observation of these connections between agricultural work and childhood cancer. And then we moved to researchers who started looking at new methods to measure pesticide exposure levels in humans and in households. And we are missing 3, and I think 3 is, I think that goes back to animal studies, and now I am, is it the, sorry, no, 3 is pilot testing of interventions. So I'm missing a block here, but 3 was where they were pilot testing interventions to change agricultural worker behaviors to minimize the transfer of the pesticides from their clothing to the home environment. And then based on that work, they were able to provide some information to EPA, who then implemented a policy that brought these interventions into standard farm worker practices in the EPA Agricultural Worker Protection Standards in 2005. The 5th milestone was that this group of researchers then evaluated the effect of this policy on farm worker behaviors and tried to get a gauge on, did the policy actually lead to lower exposure levels in the farm worker households. Milestone 6 is where the pilot test that was going on earlier actually moved into a randomized controlled trial where they were conducting the intervention in the real world. And based on that information, we jumped to number 7, which is another policy in 2015. EPA updated the agricultural worker protection standards based on information that was obtained from the data of the randomized control testing. And then finally, milestone 8 is an attempt to continue changing the behavior of the farm workers and their families by communicating the research findings back to the farm workers and their families. So as farm workers are participating in the studies and they're getting information about their levels of exposure and the exposure levels in their families, their households, the researchers are communicating that back to the study participants. So this type of graphic allows us to see where the bridging is happening, and allows us to count the number of within ring bridges and the number of bridges going on from ring to ring. And you can go to the next slide. And just to point out that this is just sort of what it looks like if we used the NCATS model, whereas before there was 1 within ring bridge and 5 ring-to-ring bridges. Using the NCATS model, there's 4 categories. And I have to say, this is actually a really lengthy case study, and so we're able to capture a lot more bridges. So that's just kind of I think the other pieces that we wanted to point out was just that words matter, because the phrasing of the NCATS translational research framework being so focused on clinical work really doesn't reflect some of the translational work that was being done to develop new methods and to communicate findings to participants. So there were some things that we felt like sort of weren't able to be captured using the NCATS model. So you can go to the next slide. As I mentioned earlier, one of the key things that the program analysis branch where I work, one of the key things we're responsible for is telling these stories of discovery and accomplishment. And over the last 10 years, one of our key goals has been to develop systems to track these impacts. Our high impacts tracking system, or HITS, imports data from our grantees' progress reports and from the notes that our program officers take on these progress reports. And then we're able to code or tag the impacts that the grantees are reporting. And we've got about 30 or so tags that we use. And you can go to the next slide. One of the things that we are really trying to do is to use this high impacts tracking system to track the translational stories over time. And this is really still a work in progress. And I actually just found out about a week ago that we just got funding to sort of develop a new module in our tagging system to create a more robust structure to capture the translational research impacts of our grantees. So when we developed this and when we started coding it, though, the ideas captured here, the things that we're able to capture here still don't include things like who was involved or in the translational story, what the time frame was, what the publications are. So we're really still trying to figure out how we can adapt this HITS coding tool to more accurately reflect the full breadth of data in a translational research story. You can go to the next slide. So the framework is a map of the research, but it doesn't really tell the whole research story. To fill in that narrative, we need more context. We've developed a narrative template tool that we are hoping will help researchers get their stories down on paper and tell their stories in a more complete way. We like to think of the map as the template as being a map that really helps them think about the path that their research has taken or might take in the future. And the template helps ensure that our grantees are using the same language to tell their stories and using a similar structure. And it also provides an opportunity to reflect on some of the aspects of the project that the grantees don't typically include in their progress reports and that aren't sort of specifically captured in the ring translational research framework in the rings. So I'm going to show you just a little bit about these narrative templates that we are using to collect information from the grantees. You can go to the next slide. The template is 3 pages long, and the first page, the one on the left, is just a picture of the full framework, and it's really just so that somebody who's writing a framework can just use this to sort of jog their memory about the possible translational activities that they might have conducted. So that's just kind of an overview page for people to kind of get their bearings. The second page, and you can go to the next slide, is set up to help outline the main points of a translational research story. So this is where you can sketch out the key milestones and kind of other pertinent sort of biographical type information. So this is more of our administrative type stuff, like who were the PIs that were involved and the grant numbers. But you can see the key translational milestones. We like to think of that as the outline, right? These are the highlights. And then for each of those milestones, so that if there's 8 milestones here, for each of those 8 milestones, we encourage the grantees to complete the third page, which is on the next slide, which is the translational bridge details. And this is where I like to think of this as sort of an interview guide. If you were sort of trying to ask, if you were interviewing a grantee and trying to get them to tell their story, these are the types of things that you might ask them. So, you would want to know, more details about what the milestone was, where was it a bridge from, and where did it bridge to, and some more description about the actual activity, some information about the time frame and the partners or collaborators that were involved. And this is also a place for them to record the citations, the publications that help support their narrative. The third page is sorry, the last section on this third page is what we call the translational narrative. It's the context. To me, these are sort of the juicy bits. This is, if you think of, if you do any active listening training, right, these are sort of some active listening prompts that you would ask. So that's how I like to think of these things. They're sort of the juicy details, but we like to get our grantees to tell us a little bit about the context of the translational story. So like, what led them to the next step? How did the idea evolve? Were there things that needed to happen in order for them to be able to sort of cross that translational bridge, and how did they know what to do next? So those are some of the things that we're trying to get grantees to think about. We're just starting to use these templates with our grantees, so we'll have to let you know how it goes. Like I said, we did just have a grantee meeting where we had about 20 grantees tell their translational stories. And I have to say, I was really excited because they seemed to get it. And it was really fun to hear them talk about their stories in a more comprehensive way. So you can go to the next slide. These are just other ways for us to think about translational research and to sort of show them and illustrate them. This is something that is on the NIH main webpage. They've got a series of stories on their page. And this just tells about the evolution of research that ultimately led to the development of a drug for a rare type of blood cancer. What we're hoping is that if we can get our grantees to start telling their translational research stories using these templates that I just described, we're in a better position to develop these kind of graphics for NIEHS to be able to demonstrate the value of biomedical research and tell and illustrate why it's important to invest in this basic research to improve human health. So if you go to the next slide, by going through the process of mapping out the translational research story for the pesticide take-home pathway that I showed you just a few slides ago, we were able to start to create a similar timeline that demonstrates the value of our investment in children's environmental health research. So this was really exciting, and I actually just presented this to some folks up at sort of the main, we call it the big NIH, up in DC, and the person who developed, who was sort of coordinating the timelines that are already on the webpage, looked at this and was like, oh, we need to get a copy of this. We want to put this up. So we need to finalize it internally and get our approvals signed off. But so it's exciting to hear that NIH is actually interested in this work. So, so the next section of the talk is, I just want to talk a little bit about our challenges and next steps, and hopefully have time for discussion. And so just to go through these quickly, some of the things that we are struggling with is really trying to figure out, is there actually any value to quantifying, to sort of counting the number of bridges, right? Does a bridge with more bridges equate to a better project? Probably not. But is there a reason that we should still count the bridges? A couple of the reasons why we might want to is to potentially identify opportunities for handoffs. If you can see that maybe there is quite a lot of bridging going on in one ring, then maybe it's time for somebody to step in and identify an opportunity to nudge that project in a different direction. The other piece is that if we can see where somebody is being really efficient at moving through the translational research framework, we can identify what strategies they're using to do that, and hopefully then facilitate more efficient, replicate those strategies and facilitate more efficient translation to public health. The other reason why it might be helpful to count these bridges is to help us fine-tune the framework and really figure out what works and what doesn't work, and to sort of keep growing the idea of translational research and working on sort of the science of translational research. Another question that we have is whether bridging within a ring, so bridging from node to node, should actually have the same weight as bridges from ring to ring. So that's something that we're trying to figure out. And then we're trying to figure out how the best way to collect these translational stories from our grantees. They're so used to telling us their annual progress that getting them to think about this long-term narrative it's just a different way of thinking for them. So we've just got some next steps on the next slide that we're doing. We're going to continue to test and refine that criteria checklist. that we use to code the narratives. We're going to continue refining and building the coding system so that we can actually have a web-based system, a structured, systematic way to track the translational narratives and the evolution of these ideas over time. And trying to develop some systems that grantees can use to submit translational stories. And then from a programmatic standpoint, because we are science managers here at NIEHS, we are looking to coordinate opportunities to use this framework to help identify and facilitate these translational handoffs. And then identifying and replicating the strategies that facilitate translational research. So we've got lots of other items on our to-do list, but these are some of the top ones. And that's it. And so I want to kind of open it up for discussion and answer any questions. And
wow, Kristi, what a presentation. That was all new for me, and I really learned a lot and enjoyed it. I have a question. So as I noticed, there is no order of coming from inner layer to outer layer, or from right to left and within the layer, or left to right. So you can jump from the very outer layer to very inner layer and just make the bridge and use the translational research within the layers without any order. Is that correct?
That is correct. Yeah, and that's one of the key things that we were trying to emphasize, because our feeling is that a translational idea can start at any point, right? It can. There may be a policy. You may have, your starting point may be a policy that's in place, and you go, "Hey, that policy isn't having the intended impact. Can we come back and figure out what's going on?" So our thought is that really the researchers can kind of draw a box around what they think is their translational story and start at any point and move to any point in the framework.
Kristi, this is Jessica Kastner. Thank you again for such a fantastic presentation. I have a few questions. One being about just one finding with clinical treatment, and with so many of us having experience in nursing interventions that are modifying the environment of the individual, as well as interventions at the public health level, that was surprising, and yet not, because a lot of that does go to other funding sources. I'd like to hear your thoughts on strategically how to bridge what nurses do and research every day and the effectiveness of our environmental health modification interventions on the individual level, and where you're trying to push some of this translational impact and measurement.
Oh, so I guess I'm trying to. It sounds like there's a lot in the question, so I'm trying to think. I mean, if I understand what you're saying, the groups that you all are working with and the work that you all do is implementing the interventions with patients and sort of affecting change in their individual behaviors. And so, is your question how we would propose evaluating that, or how we would propose sort of pushing that or helping grow that to the next phase of, through the translational framework? I guess my initial thought, without knowing, just sort of at a general level, I guess my thought would be, are there, right, if you're doing sort of individual interventions, right, part of it is
the research trial of the intervention. So it's the
right.
Science, yeah.
Yeah. So, right. So can those, right, are those findings then ultimately used to say, hey, this works, right? Hey, counseling kids with asthma to put in air filters in their, the special air filters in their homes really helps reduce. We're seeing, clinical reductions in asthma rates and emergency room visits in those households where they do the air filtering. Right? Can we move that then to a policy? To some kind of policy, whether that is maybe within a medical system, maybe it's just a hospital system or a emergency room system that says we're going to give this guidance every time a kid comes in here with asthma. Is it a policy at a community level? Does maybe a city say, hey, we see that this works and we think that we should fund a program to put air filters in the homes of kids with asthma, or right. So I guess that's where I would see the kind of work that it sounds like you all are doing, bridging to something else. I think that, right, I mean, another piece might be that as you collect the data on the interventions that you're working on, right, I mean, that's data. So you're sort of making, you've got observations about behavior. And so can you look at that, and is there, sort of moving backwards, right? Is there some question that you might want answered, right? That you can see that something is happening, right? Does that raise a question of, why is this happening, right? If you can see that, that some, I'm trying to think of an example, right? If you can see that some intervention is having an effect on, I'm trying to think, right? You do a lead prevention intervention and you see that it has an effect on weight as well, right? Do you report that back and say, oh, wait, we need to figure out, well, that would be interesting, right? To figure out why that's happening. What's the mechanistic way? What's the mechanistic understanding that we can find to explain why that's happening? So I guess trying to look at what you asked and look to the outer rings, and then, or take it maybe back to an inner ring. Is that what you were asking?
It is, and thank you. I think it's just an excellent opportunity to put on the table that a lot of this type of research is done in nursing, both at the individual level and at the community level and beyond. And what a great opportunity. You did a beautiful job sort of tying the case example to some things through NIEHS and through CDC. And then, how do we communicate then how a lot of NIEHS basic research does get translated into nursing interventions and nursing studies, and
right,
to continue that dialogue, because it's an excellent framework. Thank you.
Sure. And I think the other thing that, right, the other way that we like to think of folks using the framework, and that we're hoping that folks use the framework, is so that, right, if you all are clinicians, that you can look at it and say, oh, right, that you can recognize your role in the process, but also so that you can recognize who might be on the other side of the bridge, right? Right, and who you might be able to work with to take the research in another direction, to continue to evolve it. So that's why we kind of like to think of it as a map, and we sort of use that map imagery.
Kristi, this is Barbara. So the framework, as I see it, could also be used to help you in a lit review, for example, identify what's been done, where the gaps are, so that you can sort of map that out that way. Am I off base, or is that?
Absolutely. And I actually feel like as we are doing some of these case studies, that's what we end up doing, is really putting together a lit review.
Okay, I can see that. So it might be actually a really nice framework for students to use as well.
Yeah.
Developing their the science in different areas. So NIH, all right, so I've seen different translational frameworks, as you've said. So I'm thinking of applying, let's say, to NIA or NINR or wherever. Do you think they would be open to this kind of a framework instead of using the other one? That yours is much better.
Right. I am not comfortable giving you advice on how you do your on sort of writing your applications. Right. I mean, the advice that we generally give is, if you're right, is to talk to the program officer in those areas. I think what I can tell you is, like I said, I presented this to the Office of Extramural Research, their data policy group, last week. And the person who does sort of the impact evaluation work for NIH, like I said, the one who puts together the big NIH stories, Marina Volkov, she is using it, and she is hoping to talk to us and figure out ways to maybe get this a little more broadly distributed throughout NIH. I know NCATS is actually, so somebody from NCATS was on the evaluation advisory committee for the Core Centers evaluation that we did. She was involved in the development of the model, and she is super supportive of it. The CTSA grantees, so the folks who are funded by the National Center for Advancing Translational Science, they're evaluators for those programs. They are using the model, so it is getting out there. But again, I think if you're thinking of using it, I think talking to your program officer beforehand is a good idea.
Appreciate that. Thanks. And also appreciate your sense of humor in the presentation.
I think I read some neurodevelopmental research somewhere that you've got to bring people back every 10 minutes. So still working on that, but I think somebody had a question. Yeah, sorry.
No problem. I was just saying that thank you so much. If there is any other questions.
If anybody wants to talk to me offline, I'm happy to have a phone call or shoot me an email. Happy to talk.
Wonderful. Thank you so much, Kristi.
Sure. Thank you for inviting me.
Thank you. And I know some people will be connecting with the